So before we start the session, I request everybody to mute your system and switch on your camera so that we can see each other for some time. So very good morning to everybody and welcome you all to the 6th webinar of plant breeding webinar series, which is being organized by Foundation for Advanced Training in Plant Breeding on a monthly basis. So today we have Dr. Ganeshan, Srinivasan, who will talk about integrated management strategies for plant genetic resources in agri-horticultural ecosystem in an Indian prospect. So the purpose of these webinars are to sensitize people to know and value the importance of plant breeding, and also to create an awareness on various aspects like technology integration, policy support, regulatory overview, farmers' comprehensions, and various other aspects required for inclusive development of agriculture in order to achieve food security. Foundation for Advanced Training in Plant Breeding, abbreviated as ATPBR, is a non-profit organization which aims to promote advanced technology in plant breeding and develop qualified plant breeders who would complement and add value to the existing system. Our efforts are towards creating a conducive ecosystem where skillful organization or resourceful people work together to develop joint courses, research, and co-incubations. ATPBR organized professional training, workshop, and various awareness sessions to build required competencies of the global standard. And we also organize annual conference named Fiber Farming Conference in different locations across India. ATPBR aspire to bring the gap between university and industry by developing job-ready human resource to feed the market requirement and encourage potential entrepreneurs to succeed in their journey. Now, I would like to take the privilege to introduce our moderator, Dr. Srinivasan, who is the co-founder and director of research at Tierra Seed Science Private Limited. He's also an independent director at ATPBR. He has been working for past 53 years as a successful vegetable breeder, teacher, researcher, and development manager, both in public and private sector. He worked with companies like Adventa, Syngenta, Pioneer, and ICAR for many years and developed several nationally and internationally released varieties and hybrids in tomatoes and okra. He has published several research papers in peer-reviewed journal and also supervised MSc and PhD student. So I request Ikusa to take the floor. Ikusa, over to you.
Thanks, Aparna. Thanks for that introduction. It became long, but good morning to you all and welcome to this 6th lecture of our series. And it's my proud privilege to introduce to you to the speaker of the day, Dr. S. Ganeshan, who also is a old friend because I also come from the same ICR stream. He was principal scientist and head plant genetic resources at IHR at the time of his retirement. He is an ARS scientist from 1978 stream and 35 years of research and research management experience. He was a visiting scientist under FAO UNDP project on tropical and subtropical fruits to UC Davis and National Center for Genetic Resource Preservation, Fort Collins in USA. He has also been on a deputation to as director, Jawaharlal Nehru Training Tropical Botanical Genetic Resource Institute in Trivandrum for 3 years, to 2005 to 2008, nearly 4 years. And he created a lot of good science during his reign. I will just name a few. He has lots and lots of good science that he has created. Created in vitro conservation facility at IHR, active gene bank that got for grapes, fruits, and medicinal plants. And I think he has got a Limca Book of Records for having created that. And he has also developed pollen cryopreservation technology for horticultural crops that led to the establishment of a cryobank for fruit, vegetable, ornamental, and medicinal plants at IHR. And I must say here that commercially, that cryopreservation of pollen today is used by the seed companies. I think in 1992, I got in touch with him and I joined Pioneer, and I think that has now become a technology that a lot of seed companies are using. I mean, seed production companies are using, where the male plant is now secure in a secure one place, and they freeze the pollen, and then the pollen is distributed to the various female plots, and that way one parent is totally secure. So it's a very good technology, and I must congratulate him for having done that work long back, and it has commercial, application. He has also 85 research papers in journals of repute, has served as consultant in Malaysia and resource person for India for the International Plant Genetic Resource Institute of Rome, where he was also a visiting scientist. He has chaired a session in Third World Botanical Congress in 2007, has been associated in various advisory bodies at national and state levels on germplasm and cryopreservation, and has been honorary editor for several scientific journals like Biodiversity, Palynology, and Current Science, etc. I think there's a lot, and I think that sums up Dr. Ganeshan, and I now give the floor to Dr. Ganeshan to let us hear him. Thank you. Oh, you are
So
on
you
mute.
have to unmute.
You are on mute.
Unmute Ganeshan.
Ah, yes.
Good morning, everybody. Are you able to listen to me?
Good morning. Yeah, we can hear you.
Yes.
Yes, we can hear you.
Yeah, we can hear you.
Very good Suprabhat for you. A very lovely morning in Bangalore. Rain, drizzled rather in the morning. It's climate is very salubrious, and those who are from Bangalore may be really enjoying it. And I, at the outset, would like to thank the organization ATPBR for inviting me to provide a talk on my experiences, rather my travels, I would say, on genetic resource management. Now, without wasting much time, let me just go over to my presentation. In case you are not able to see me on the screen, please let me know, because I keep moving while I am presenting. At the same time, I wish to be around and ensure that I show my face so that, we are, yeah.
Yes, we can see the presentation.
Yes, we can see your presentation as well as you, both.
Thank
Put it on
you.
slideshow.
Yeah, I'm doing that.
Yeah.
Okay, work
Perfect.
fine. Perfect.
Yeah.
Thank you. So here we go. So the talk of today is based on management strategies, how best you can integrate them for genetic resource in agri-horticultural ecosystems, where I would say that, right from 1977, I bring to the knowledge of many of you that our founder director, Dr. G.S. Randhawa, initiated me into the work on genetic resources conservation. From then on, I have been fortunate enough to work for almost 3.5 decades to 4 decades in the area of genetic resource conservation and management. So, genetic resources, what are its origin, diversity, and use? Genetic resource, in our breeding parlance, the meaning is entirely different. When you talk of genetic resource to another person, collection of plants are synonymously thought of as collection of genetic resources. And this misnomer is always being carried out, even in one of the international programs where many people were telling that I have added 10 accessions and 20 accessions to my genetic resource material. I said, absolutely wrong, because genetic resource is something different from whatever collections you already have in your kitty. So when you talk of the country's rich species biodiversity, it is one of the 12 mega centers across the world, and it's blessed with 2 mega centers of biodiversity. One is the Hindustan Center, the other one is the Central Asia Center of Origin. Now, we all know the Vavilow's theory of center of origin and center of diversity, which we studied in our plant breeding class. The biodiversity is mainly distributed in the Western Ghats, northeastern India, central Himalayas, and from where the indigenous plant genetic resources are sourced. What I mean is the origin of your genetic resources come from the biodiversity. And many species originating from the tropics, subtropics, and temperate have rich agri-horticultural value. When I say agri-horticultural value, I just keep set aside the agriculture component of it. I speak more in favor of horticulture because I have a bias, having worked there for nearly 4 decades. The vast degree of diversity present in the Hindustan region is definitely and directly related to the high divergent ecosystem and altitude variations. So the Indian scenario, continuing about it, we are very floristically rich. We have about 141 endemic genera out of 5,150 species. This may be a bit early data. I think this was collected around the mid of 2015 or 16, 14 or 15. Compared to China, China is almost having 1/3 of their plant collections endemic in their biodiversity. 1/3 is a very big number. And here they belong to 47 families of higher plants. Among the endemic species, we have 2532 species that are distributed in the Himalaya. This is the temperate material. 1788 species in the peninsular region. Very, very rich in diversity. Then 185 species in the Andaman and Nicobar. In fact, we have replicated in the Jawaharlal Nehru Center of Tropical Botanical Garden 120 species from Andaman and Nicobar, and we have an Andaman block in Trivandrum. If any of you are likely to go there, you can visit it. There are about 43,000 plant species that are said to exist in India. Maybe now it may be slightly more because there was a revision of the Red Data book, of which 7,500 plant species are referred to the Indian folklore. That is, the medicinal plants which have high values, which we do not know. Somebody else from another country would come and identify its properties and take it over and then make the final product and sell it back to us with a higher price. So this is the problem with many of the folklore medication, which we are not in a position to identify its genetic resource or the traits that are present in it. About 1,700 plant species are actually documented in the ancient literature. I'm talking about a literature which are on the leaves. We did have a project in Trivandrum where there was a program to translate information from the leaf document into digitizing of the entire information present in the leaf documents. So these kind of activities would certainly enhance our number of species that are present, and where exactly they are present needs to be checked out. I mean, we have to make a clear-cut demarcation as to where we stop and where we start the agro part of it. So genetic resources derived from these group plants calls for an efficient management and economic use. Given the emphasis during the preceding decade, as designated as UN Decade of Biodiversity. We all know 1020 was the decade of biodiversity. So agrobiodiversity derived genetic resources, we talk about that. In India, it is highly distributed in 8 very diverse phytogeographical and 15 agroecological regions. The range of distribution of these plants varying from, these are the 4 areas which I have listed: evergreen forests to the Western Ghats, then deserts in Rajasthan to mangroves in the east, from the vast deciduous forests of Deccan Plateau to the sholas, higher near the area, that is the foothills of Western Ghats, from the swamps of Ganges, that is the mangroves, to the moss-laden tree trunks of the Silent Valley. So agri-horticultural cultivation practice, as you know, even before we thought of genetic resource, before we even conceived the idea of germplasm, have evolved from time immemorial, using agrobiodiversity sourced from these regions. So horticultural crops in India, when we talk of them, we have a high value and low volume. And many of them are indigenous, say about 70%, which calls for re-examination of our country's biodiversity. Then exotic is to the extent of 30%. Then cultivated in all agroecosystems. We all know that horticulture is present everywhere, distributed in all plant forms of habit. Then that is herbs, shrubs and trees, perennials and all kinds of habitat is present. They require diverse management needs, as we all see it, because it is distributed in all kinds of, all forms of habitat. It requires diverse management needs. Emphasis mostly on field gene bank and clonal repositories. This is the major focus which normally plant genetic resource in horticulture is given. So the distribution of crops in the agro-ecosystems, I am just listing out what has been done in the NARS. Maybe this list is not exhaustive, so or maybe exhaustive. Different fruits and vegetables and a number of crops which we are right now dealing in these different categories. And what is the distribution? Tropical, subtropical, temperate, arid, semi-arid. And sometimes, new species are discovered by the Botanical Survey of India, which is in the form of a vegetable, and that can be also identified in one of these agro-ecosystems. Then what is the crux of management here? Focusing on the management part of it. We have to identify genetic resources of relevance to Indian agriculture and horticulture. It's not that you can separate these two here, but something will be useful in agriculture, something will be useful in horticulture. These come from wild species. Very well-known wild species in field crops have revolutionized agriculture. Similarly, genotypes, individual genotypes. Like I remember that particular one in that kiwi fruit, which has come from China, and that has completely revolutionized the cultivation of that particular fruit. Then extant crop varieties. We all have many of the varieties which we have not been kind of documenting, but still present, which were more than 50, 60 years old, maintained by farmers over generations, over families. So knowledge of genetics and principles of plant breeding, though not was aware with the farmer, is now essential to classify the plant genetic resources and maybe perhaps make it in a more inclusive manner. Take all stakeholders into account. So the types of germplasm which we have categorized are these 12 germplasm we have identified from the breeder's perspective. And to this, someone has to tell whether we should add the GMOs. There was a lot of debate on that, whether it should go in this category as germplasm category, which are not being right now used in cultivation. But do they get a status of germplasm or genetic resource to be categorized under this kind of a setup? So just giving you an idea as to how much of germplasm has been registered in these crops. Maybe a bit old, but then I don't think there will be any enhancement in this list unless Dr. Bansal in his talk would have made a latest presentation about what is the current status. Now let us deal with the components of genetic resource management in a cascading activity format. That is, initially we take up a survey, exploration, introduction, domestication, regeneration, characterization, evaluation, conservation, documentation, and distribution. But this somehow sums up all the activities in genetic resource management. So let me first take a survey, whatever little bit of experience which we try to do at our institute. We use this mapping software, RSGIS software, and there was a document, a compilation made by Dr. Ganeshan and his group called as Jiva Sampada. We use their software and we tried to map some of the species in the Western Ghats in one of our projects where we had to identify species of horticultural importance. So we were able to do it for this many number of species. I hope we are able to read them. They are a bit smaller because I took a screenshot of the screen, TV screen, computer screen. And more details can be provided under each folder. If you can see the details, I'll just take you to one particular folder. In this, we have done some sort of a distribution in the Western Ghats in two states, Karnataka and Maharashtra, where the distribution map is showing those grids. We use the grid format for locating the species. And you can see the map here with different intensities where the diversity is directly correlated to the colors that are being shown here. And some of these things, which are the states that are covered, distribution data in so many places that are covered. This is for Garcinia indica. And we have given in the flyer, the picture, the entire taxonomic details and distribution in different locations. And we also have the lat long information It is not present in this flyer, but they can always go straight away to that location plus or minus some 10-20 meters, but try to locate this particular species for collection. So this has been a very successful program which we were able to do for at least more than 15-20 species of horticultural importance. Now, when we talk of survey and exploration, we also have to think of the gaps and missing links. Strong collaboration between various institutes of working in horticultural crops with the national centers, that is Plant Genetic Resources Institute, National Bureau for Exploration of Horticultural Crops. Intensive explorations are needed, similar, preceded by surveys covering all horticultural crops, especially where collections are less. We all know that, in all crops, we do not have adequate germplasm collection to represent the entire genetic diversity. So that is the main reason we say National Exploration Plan should focus on all crops. Crop breeders in horticulture feel that their exploration needs are not prioritized, are not to be prioritized. Whenever this particular discussion comes, I have been attending in meetings in NBPGR, field crops always take a preponderance over horticultural crops, although today we have surpassed the yield levels of field crops. The timing of exploration is often very general and not specific. And proper record keeping of exploration missions, we have been doing this right from the National Agricultural Technology Project period. Documented information has been supplied to NBPGR, and we have been giving enough inputs in horticulture to NBPGR as a center working on this genetic resources. So when you go again, explore and collect the trait-specific germplasm and wild species relatives of horticulture crops. Now again, how do you go and do this? Trait-specific germplasm. That should have a very good breeder's eye. In those days, breeders used to go in the field and spend hours together to identify a trait, maybe visit and revisit several times. I don't know any scientist today has that much time, but this is inevitable for a breeder to look for the trait-specific germplasm, at least the phenotypically recognizable traits can be thought of. Then germplasm collections having high nutraceutical value. Now, this is another area which we have been lagging behind, especially leafy vegetables. I know very well, in front of my house, one lady comes and sells leafy vegetables and, wrapped in a very shabby way and not very, presentable. But in our country, we keep our chappals and shoes in air-conditioned rooms. But these leafy vegetables which provide the nutraceutical value are thoroughly being neglected. So a group has to really take lead and do this one in order to strengthen our country's, health overall. So georeferencing hotspots using remote sensing and GIS, geographical information system tools, before taking up exploration mission. This is very, very important. Without georeference data, you're only going for a pleasure trip. So this also has been many a times reiterated in meetings, and many of them don't want to invest on this kind of an activity. Anywhere there is an exploration program going on in the Northeast, how many would like to join? So everybody go, collect whatever is available and bring it. And say that I made some collection, but you have only collected the plants. But what is the diversity that you have collected? Nobody knows. Plant crop specific surveys in centers of genetic diversity, cutting across political regions. This is a very important aspect, especially with Biodiversity International being present, showing its presence in India and also in Bangalore. Institutions will have to take up coordination activities, cut across the national boundaries, have some sort of an arrangement like agreements and things to facilitate scientists, breeders to go and look for the material there. Then planned exploration missions in consultation with crop-based institutes is very important. Repeat introductions, like many a times we have seen a lot of material brought in repeated, but there is no way to really check. Maybe a molecular method only can be brought in place to do this. Multiple exploration missions for collecting material at different growth stages, especially in case of perennials. Exploration and collection missions from outside India, which I already spoke about. Then distribution status, exploration and collection. When I showed distribution status in the form of maps, where do you see and how many years back it has been seen. This is a continuous activity. And collection of native land races and geographical indicating status and farmers' varieties in horticultural crop not fully systematized. There is a need to shift from genotype-based exploration and collection to diversity-based collection. Strategy for minor fruits and tribal vegetables, spices to capture diversity for nutritional needs I've already discussed. Planned exploration to collect native diversity of indigenous ornamentals. This is a very, very important area. Today, our ornamental industry is flooded with exotics. We have very good ornamentals, indigenous ornamentals, which we have never been thinking of bringing it to the fore for cultivation. Then there should be a gap analysis for exploration and collection of wild species based on taxonomic identities. This is a very important thing. We have been neglecting taxonomy. I remember Dr. Mohan Ram telling, who will take taxonomy? It is tax on me, he used to say. So, coming to the introduction part, like, in many of the perennials, fruit tree species, plants which are introduced into a particular ecosystem, brought from the forest ecosystem, do not survive, or survive only to the extent of the vegetative growth. But many a times we do focused introductions for the sake of breeding work. One such work which Dr. Ganeshan at ICAR-IIHR, who works on mango, rather just retired a couple of months back, he was struggling to bring in Camptosperma from the Andamans and plant it in ICAR-IIHR in the Asarghatta region. Unfortunately, several times these attempts were made over one and a half decades, but it didn't work. But then what thought came into his mind? Grafted Mangifera indica plant with Mangifera Camptosperma scion. We brought in one of our explorations to Andamans, we collected the scion material of Camptosperma and grafted it on indica. Very clearly you can see the fruit shape and size which represents Camptosperma. So the grafted material has been successfully introduced and we are able to maintain this in our field gene bank at Asarghatta. So this is one of the success stories. Similar things can be replicated for other perennials where there is a problem of introduction in the form of plants. And this is another one which I thought I should show you. This is this Bakuria cutralensis, or called as kattafal, a very well known plant in the southwestern Ghats, and many of them are being used by the tribals. Tribals use it for its high antioxidant value, vitamin C value, flavonoids, and so many attributes are there for this. But it bears copious fruits in the trees. And at the time of fruiting, it's a sight to see. And this one we successfully were able to introduce into the Indian Institute of Horticulture ecosystem. But I tell you, I have failed to induce flowering, and it grew for 4 years, but it never induced flowering. We were able to multiply enough number of plants. Don't know what happened after I left, but a lot of material was generated. And again, priority kills this kind of activity. The priority is for those crops which have immediate requirement to be multiplied. The land, providing the land for multiplying this or growing this individually, we may require at least half an acre to 1 acre. But production availability of this for putting this, transplanting these plants was not made available, and we couldn't plant it. But wherever we were able to successfully do it, we did it for this governor's plum, Flacosia montana, and a field gene bank was established, and it is now still standing there in the institute. If you are able to go there and see, then we were able to see the dioecious flowering in Flacosia, and dimorphism also in the form of thorny and thornless trunk. These are some of the traits which you can visually observe phenotypically. And this is what, when I say that when you go for exploration, you have to look for these kind of traits, and then maybe initially you may feel this may or may not be useful to us, but you do not know how many other valuable traits these phenotypic traits are linked. That has to be understood. So introduction, the gaps, missing links, most introduced are received late. Our introductions, it goes through all the channels. bureaucratic channels and government of India route quarantine. And sometimes it is even beyond the timelines of a breeding program. So there is a need for a curator, like you have a curator in the field crops. Breeders often feel that there is a delay in receipt of the material from NBPGR to the breeder, and when it comes to him, breeding time is over and someone has to maintain it. There again the problem is perennials have a problem. It requires proper curation. Then while introducing the germplasm through exchange, the strategy for exchange policy issues are not put in place. There are a lot of problems when you talk of exchange, when many of them are not even willing to exchange. There is a lack of proper acknowledgement of PGR introduced and used by breeders for developing cultivars, because they take it for granted that it is a plant genetic resource. From there to the release of the variety, everybody sees the retailed variety, but what genetic resource has gone into it to make that variety, there is a very less acknowledgement provided. The introduction of genetic resources is not happening due to IPR issues. Recently, this has been a problem. I remember very well in the 80s, we used to get material, at least for no efforts made. No efforts are made to strengthen the ties in the donor nations. This I have been trying with Bioversity during Dr. N.K. Krishna Kumar's period to have some tie-ups with countries like Iran, Iraq, and we were able to get successfully maintained pomegranate collections from UC Davis, from where one Dr. Aradhya was kind enough to spare the material for our pomegranate work. There were a good number of accessions from UC Davis. Of course, these are secondary collections, not from the primary source. Then trait-specific germplasm, core sets and wild species are not imported or introduced to the desired extent. This is not happening. Here is where I see the role of private seed companies which are distributed across the world, where they can come up with some sort of a network program and dovetail it with the country's requirement. No proper care during import of horticultural products from certain countries leading to infected material. Yes, this has happened with my grape material which I tried to introduce from Germany. We got about 28 to 30 accessions as cuttings, but when we finally were able to take it to field, I got only 14 or 15 of them surviving. And there is no attempt made to introduce new morphotypes of ornamental genetic resources for commercialization in competition with the exotics currently ruling the market. There again, when we say introduce new morphotypes, these are some of the material which have become extant, where there is no IPR issue. Such material can be introduced without much problem, but this is not happening. The lack of intense efforts to collect and introduce material from secondary sources, which I have already told you. Then coming to evaluation and characterization, there is a shift. There is a shift to total morphological end-to-end characterization. This is very important, which is not happening. And now what is presently being done is breeding objective-based evaluation characterization. The breeder has, say, n number of germplasm. He looks for only evaluating and characterizing those traits which he wants it for to fulfill his breeding objective. This molecular genomic characterization, species-specific traits, marker trait associations, and diversity analysis has to be taken up. Pre-breeding to form core and mini-core sets. These are available in very, very few horticultural crops, but not being pursued actively in most crops. So what are the priority areas? Evaluation of biotic, abiotic, and quality seeds quality needs to be carried out for minor and temperate food crops. When we say minor, somebody has put it as minor because there are already a dozen major food crops for which we have been prioritizing our work. Cucurbitaceous vegetables, medicinal and aromatic plants, these are all being relegated to low priority areas. Mini core available for a few major and temperate food crops, and also a few vegetables, like in Dr. Keller in Gartersleben had a mini core and mini core for onion and garlic. Of course, now he has retired. He was one of the persons who took my technology for pollen, and he has established a pollen bank for alliums. And this needs to be thought of, where core and mini core sets can be made available and exchanged with whatever we have developed. the need to be carried out in vegetables, medicinal aromatic plants, we have mentioned about. Pre-breeding activity, which is seldom carried out for fruit, medicinal aromatic plants. Sorry to tell this, the breeder in fruit, medicinal, all these crops, just grow A and B and collect pollen from A and cross it with B. And maybe he has a set of, first of all, he has a very narrow genetic base to operate. So from that, he doesn't know what characters are going to come through into the desired variety which is developing. And this is something like hitting a pot shot. So this has to be kind of streamlined to the extent possible, whether he is likely to generate in which generation the concerned desired genotype which he is looking for. In major tropical fruits also, low emphasis on genomic and trait-specific molecular characterization to develop core sets. Well, yes, we did have a project in mango, teamed up with an Australian group. And this work, I don't know, at that time this was being carried out. ICAR-IIHR is one of the partners. How much it has yielded the results, only someone can Google it out and check it out. And only, when after we make several times, we tell them this kind of activities have been funded in horticulture. In temperate crops, minor tropical and subtropical fruit characterization evaluation requires priority. In all these crops, there is less priority. In vegetables, gourds and melons, characterization and evaluation, pre-breeding requires priority. Spices except ginger and turmeric. There has been some activity to develop core sets by ICAR, and we have to use all the tools to prioritize these activities in these crops. So, performance in different agro-ecosystems. This is being done by the AICRPs at various centers. I happen to attend one or two meetings of there. Then they also have taken up some morpho and chemomolecular characterization work for specified material, promising material I would say, because it is a cost-intensive technology. So from PGR to product is another aspect which I would like to just give me a moment. I'm in a meeting, I'll talk to you later. So, development of trait-specific PGRs for value addition. This is something which we need to think of. And I was talking about PGR to product here. Annona muricata. We don't know anything about its genetics. We don't know how much of it is being bred across in the country. But we know very well that, this milk from Annona muricata is used for, some carcinoma. So to cure some kinds of carcinoma. Even some doctors have accepted it. But right now, what is happening is this is being sourced from the wild and directly product is made. And this, what is being done by many of the NGOs, we need to kind of streamline it and give the support to the tribals who are providing this material without, of course, destroying the diversity of it. So, the gaps that are there, I have put up so many kinds of information. I think I'm running short of time because I have only 5 minutes. If there is a possibility of extending by 5-10 minutes, I can cover. Total evaluation characterization is seldom attempted. No attempts to characterize and evaluate germplasm. Lack of participatory programs, and there is no protection for characterized PGR. This is something which is very important. We need to protect, give a value tag to characterized PGR. Otherwise, it becomes a problem for us. Tomorrow, somebody else would be taking the material for utilization. Then, again, I have listed out several such missing links and evaluation in characterization evaluation. So, these may be looked into. In many times, we are facing many of these issues in horticultural crops. There is no
Please carry on. Don't stop. You can carry on for a few more minutes extra. No problem.
Okay. In conservation part, now I'm coming to the conservation part, of course, this is an area which many of you are familiar, and what is being happening in India with regard to conservation, and these are the different formats that are present for conservation, and at what level we think of conserving XC2, that is genotype, organ and gene level. Then we have these approaches for different gene pool components. Then we have, unlike in field crops, we think only of seed. In horticulture, we have to think of seed, in vitro, pollen. All the categories have to be thought of, including field gene banks. And this I have tried to put it to show the diversity of conservation methods. This is the field gene bank which we established for RET species. RET stands for Rare, Endangered and Threatened Species. We had a project with one National Medicinal Plant Board, wherein 10 centers were asked to choose 3 to 10 species, and the objective is to see to it that these are removed from the red letter data book, so that you increase the numbers and then exchange. So each center in the end of the project will have at least 100 species with them. And this was something which we have done very successfully. And I thank Dr. H.P. Singh for having given support for this program when he was chairman of the National Medicinal Plant Board. And this is some of the, just to give you a picture of a gene bank in across in a typical gene bank and how things are being stored. Of course, these are very well known. Then we have this in vitro conservation centers where we have been able to maintain material in slow growth, in slow multiplication, and using minimal medium and using some osmoticum and things like that, and managing to retain its growth in the tube for several years. This is primarily a backup for field gene bank, thought of as a backup for field gene bank. Whenever there is a plant dying in the field, we need to think of replacing. This is successfully done for cassava and banana also. And of course, we talk of cryopreservation as a holistic thing. Of course, I have done a lot of work on pollen, and we really understand the problems when it comes to vegetatively propagated species. Successfully, it has been done for hoa, coconut and coffee, for recalcitrant seed, for roots and tubers, for vegetatively propagated ornamental fruit tree, for medicinal plants, for alcohol producing cell lines. We can maintain cell lines indefinitely. Then transformed tissues, genetically transformed tissues, transformation, mutagenesis, competent tissues. Then removal of viruses is very successful in banana and plum when you store the apical meristem. Then conservation of plant pathogens is another area coming up, and we have a National Institute of Agriculturally Important Microorganisms. I think it has been shifted from Maubanjan to, I think, some other place. But NBPR is still giving a vault to them. So some outreach activity regarding the cryopreservation in which we participated, and we were able to successfully train a good number of people in Southeast Asian countries. Then we have this picture of what is existing. I mean, this is a bit old picture because the National Seed Storage Laboratory has been renamed. And what we have been doing at IHR also, I'm just trying to show you. So just to give you an idea, we were also involved in getting this in the Limca Book of Record, and we started our pollen cryo bank in '83. So there's a DNA bank which I could capture a picture of it in Korea. So it is something which just is coming up, but how far it has been successful in mainstreaming activity of genetic resources, I'm a little bit short of knowledge. So lack of adequate institutional policy. This is something which we should keep in mind. Operational management that are fair to all stakeholders. Decisions on management of genetic resources made in isolation, not as an integral part or a component of a national plan which is to be mainstreamed with. Then we have what I mean by stakeholder is interest holder analysis to be done to identify pressure on resources, land use disputes. Who will be affected by PBR activities? What are their interests? Who have the right to participate? How stakeholders can participate in different PBR activities? Calls for inventory of different stakeholders. And I have listed the stakeholders. Some of these people may be irrelevant when you straight away come to the look of it. Okay, so this is how I classify them. Then thinking of priorities for stakeholders. Stakeholders look for economic, religious, traditional, spiritual, and aesthetic, and priorities may vary for PGR management. And their interests are also entirely diverse. So if you go and tell a person in Kerala to keep up a jackfruit tree and a mango tree and tell him that this mango tree is useful for me for my genetic resource, he will see the economic value for it. He will see this is unnecessarily occupying space. If I cut it, I can get a lot of firewood from this. So he looks at it from that angle, whereas we look at it from a genetic resource point of view. So these interests vary. So there's a conflict of interest. So we need to do a lot of capacity building, which requires participatory processes in management. Communities often to be strengthened for their organizational capacity, made better capable to reclaim responsibilities in management of PGR, identify intrinsic skills in communities. Then development of competence, such as practicals in keeping plant records. We talk of all stakeholders. I'm not talking only our own scientific community. Obtaining training in technical, and we need to kind of extend our hand as an outreach program. Training in mapping their own land races. This is something very important which I find, because when you go to an unknown area, the local farmer is your guide to take you to the place exactly what you want. Training is training in conflict management, resolution of disputes related to PGR management, impart capacity to communities to gain user rights, acquire skills for financial accountability. These are all people, many of them may not be as educated as us, breeders, cooperate and develop ways to ensure natural plant resources under their control and management. If you give them empowerment to maintain the material, we can be sure that they will definitely give you good genetic resource material, or maintaining them in situ. Encourage custodianship. This is a recent development which has been conceived by Biodiversity International for mango, and this custodianship has really worked very well in maintaining many of the tree species, so that better organized outside interest groups will not overpower and take over. So the ability to scrutinize the intentions outside investors and develop. This is something very important. These people have to be strengthened for such scrutiny. Reject outside interests if these are not beneficial to the community. We breeders do it. If something is not of our interest, if it is not within the objective framework of our breeding program, we don't want that material. So similar knowledge has to be imparted to the other stakeholders. In areas ranging from basic education to reading, many of them are illiterate, they don't know arithmetic. And training in planning of PGR activities, some of the agencies which have thought of, which can be involved in these kind of activities I have listed. Then approaches to management need to be based on participation of local communities. Impart capacity building to maintain ecological processes that support conservation of ecosystems mapped with high levels of genetic diversity. I remember Dr. Madhav Gadgil's activity, and his entire report was turned down by the government. Approach developmental goals to meet plant genetic resource needs for future generations, besides meeting their own needs. Then issues which need to be addressed, I have just listed out. I'm not reading them. It can be just read out by on your screens. Awareness among user groups. Then resolving conflicts, very important aspect which we have seen. In many cases, it has happened between breeders and the farmers. Then prioritize for large-scale in-situ maintenance. This is what I said. When you give custodianship, you can give custodianship for large-scale in-situ maintenance, promote location-specific plant types and associated species. Species-level categorization is important as well. We discussed knowledge of good post-harvest, other associated domains. I just brought this into picture when we talk of PGR, especially PGR to product. Post-harvest technology definitely helps to identify the product in a very good and formal manner. Then encourage knowledge centers for PGR by teaching communities. This has been proposed by Dr. M.S. Swaminathan in his own activities, in the tribal activities in Orissa, where he says conservation is a continuous process. And we say conservation continuum should be pursued. Then way forward, identification of genetic resources in species ancestors. What is it that ancestors can provide us? Research in genetics and breeding, increase availability of core trade approaches already been discussed. Promote a system of national prioritization, and with different kinds of crops, the networking mode. There's a need for a comprehensive relook, call for a deviation from conventional PGR management, conceive a set of new normal guidelines, promote national and or collective efforts to conserve genetic diversity of species which are regional, national, global importance. Then promote many of these issues which have been talking earlier also. Some of these are getting repeated. Then here I would like to focus in safety duplication of ex situ conserved genetic resources in a place like Chandra or Svalbard, where we have already planned it up with the NPPR, already taken up this kind of an activity. And utilization of genetic resources is a very important aspect to develop innovative products and procedures. Product development directly from PGRs is immediately needs for food, nutrition, and healthcare without depletion of natural reserves. So, lack of adequate collection, domestic measures to identify and translate, express species inherent traits among genetic stocks, lack of studies on trait-specific inheritance patterns in PGR of most crops, the requirement for pre-breeding, and how to enhance availability, identification of purate types, identification of characterized germplasms, use of trait-specific pre-bred core sets. The other day I was hearing Dr. Tonapi, he said only 3% of our genetic resources are being put to use. How are you going to think of enhancing them? So, coming to almost, I think I'm left with 1 or 2 slides. Web-based permanent, many permanent problem. When I say permanent, a worker in PGR has to work for decades. Like I worked for 40 years, but if somebody gets a higher position, he leaves the job, he doesn't get publications. If he's a very great aspirant in climbing up the ladder, then he will not stick on to PGR work. Strengthening multi-locational breeding activities involving core sets of PGR, then reviewing policies impacting horticulture. Then on-site constraints have to be thought of. Bureaucratic obstacles for use and distribution of the genetic resources. Dr. Tikku, you may be very well aware of all these things. And sharing of responsibilities, the NPPR is the only nodal agency. Now we need to conceive a national institute under NARS to coordinate and intensify horticulture PGR management activity. So we need to think of adopt Green Global or GenSys, which earlier IPGRI used to use it, or Biodiversity now. So bring a complete set of continuous monitoring and comprehensive monitoring is required and assess threats to target species and establish a system of monitoring genetic diversity on long-term basis. So this is the last slide. Exchange of experiences using information technology through capacity and build capacity through organization of joint regional courses and workshops. And development of knowledge and expertise to facilitate harmonized genetic resource strategy, policies and legislation, build strong national participatory programs with all the public and private institutions, then promote sharing of PGR. This is very important between public and private institutions with a robust benefit sharing mechanism. Thank you. I think I've exceeded by 9 minutes. 9 minutes.
Thank you. Thank you. That was excellent timing also. Thanks a lot. Thank you. Aparna, I continue? Aparna, are you there? You can stop presenting now so that we can have. Can you stop presenting?
My presentation is over.
No, you stop. You have to stop presenting. Then the screen will be. Yeah.
Yes. Yes.
Thank you very much. I think the participants will appreciate that you covered a lot of ground. I'll kind of moderate, and I thought before that we have some questions on the chat box. So, Suresh, are you there? You want to ask directly?
I think, okay, sir. Thank you very much. It's really knowledgeable and informative presentation to understand the various genetic resources situation in India, and how it has been conserved and under different management approaches. I also see a lot of concerns which he felt that he has raised, mainly in terms of when the genetic resources is shared. What are the multilateral agreement systems, especially when it is shared across the international organizations? What sort of arrangement? Because it has a lot of implications. Number one, that access benefit sharing, when people uses the genetic resources, so they need to share the benefit what they gained over the years. So otherwise, the resource cannot be mobilized to build the future genetic resource conservations. So what are those dimensions which India has developed the policies across the different the genetic resources? Number one. Number two, whether only the genetic source or the knowledge is also associated while sharing, because sometimes people get the genetic resources, but they're not knowing what are the knowledge behind the genetic resources, especially medicinal aromatic plants and many other horticultural products. And how about its genetic materials also is shared. Sometimes DNA fingerprinting, they may not know how to protect those genetic resources. Number three, especially the rehabilitation is also important. When we take the genetic resources from the villages as a land race, so we improve, and also sometimes we need to rehabilitate to help them, show them how it has been improved over years, so that it can be taken long-term approaches.
Okay, I mean, let us, Dr. Ganeshan, did you get the question?
Yeah, I got the questions, but I don't know who the speaker is.
Dr. L.M. Suresh, can you put on your video?
I'm not able to see it. Yeah, I'm able to see it, but
Suresh, can you put on your video? Yeah, here.
Yeah.
This is Dr. L.M. Suresh. He works for CIMIT based in Kenya. He is a very reputed pathologist and does a lot of breeding for disease resistance in maize, etc. there.
Okay.
Yeah. So he's an old colleague and friend. Yeah. He is not old, I am old.
Thank you for joining Dr. Suresh, and although this may not be your direct domain with the present work which you are carrying out, and I really appreciate your concerns with regard to genetic resource management. What I have presented here is a bird's overview, or rather a glimpse. I know Dr. Tikku will definitely bear with me. Many of the issues which you have asked are still yet to be sorted out. Even I think few seminars before Dr. Bansal was here, Dr. Tikku.
Yeah. Then Dr. Kuldeep Singh also was there.
Yeah. So when these people were talking, they would have given a much later information about all the policy issues, but I do not know whether they are covered. But when it comes to horticulture, it is always the lame excuse. Many a times I have seen that, our breeders criticize in horticulture, criticize NBPGR for their untimely or not providing germplasm on time. Yes, you are right. The benefit sharing aspects are required to be worked out, and this we have been putting a lot of emphasis in all our AICRP meets also. Dr. Tikku may be able to, support me or give me an additional inputs if you feel like. But many a times I have seen my personal experiences, what you people are doing horticulture work, it is not kind of, honestly I tell you, I don't know whether there are NBPGR scientists in this group. The budget for plant genetic resources, 80% is taken away by field crops. Now what percolates to us is left over. And we have seen this many a times. We have made efforts to, in fact, during my service period, I made an effort to move a National Center for Plant Genetic Resources for Horticultural Crops. This proposal is still lying in ICR, if I am correct, but again turned down because they say for the country there should be only one genetic resources unit. But today we have grown beyond. Try to understand, we have surpassed field crop production. The number of crops which we have in horticulture is almost or equal or slightly more than field crops. I don't know what is the number.
They have much higher economic and cash value. Horticultural crops.
Horticultural crops have a lot of cash value.
Huge value. Yeah.
No, I remember very well, Dr. Sawant wanted to introduce a lot of grape material from, I think, France or one of these countries. He was completely, being told that, it should not be imported, it's not possible. But then he had to use some of his political channels to get the material because he was facing a lot of pressure from the grape growers in Maharashtra. To be very frank with you, maybe I may be talking a little out of the box, but the situation is not all that rosy for horticultural crops when it comes to import of germplasm.
Yes.
And many things have come, but they have not reached the concern. And I remember Dr. Sawant has told me that you have to go sit in front of the office, sit in ICR, and then get it cleared. And Of course, little bit of political intervention he had to do because the pressure was more on from the farmer side, grape grower side. Now, such a thing can happen to many of these kind of horticultural crops where the growers are a bit ahead of us in getting the material planted in their place. Anything
But
else?
only,
There are a few questions. Anything else?
yeah, only as you said that when you want to take from the international organization, any international organization, they always have kind of a international plan treaty like SMT agreement. They normally attach to it. There should not be any botherations unless India is not part of it treaty.
Sir, this material transfer agreement, I did it with several countries when I was in service.
Yeah.
And through that only we were able to get some material. But at some point of time, that also did not work out. Because when the helm of affair people change, they have a different thought. And these are normally for a restricted period of time and needs to be renewed every year. This renewal could not happen.
And also this mutual, if it is not mutual, then it kind of stops. It cannot be one way.
Yeah.
That is an aspect we have to look at. Of course, there will be IP issues, etc. But if somebody is giving us something, then they will also expect something back from us. Germplasm, without that, I want to give an example that we would not be eating tomato and corn in India if it had not come from South America, right? And there are so many crops we have also benefited, and this should be there should be work out of. I think it is part of the that UPO meeting, etc. They had worked out a lot of methodologies, but how far it is in the system now, I do not know. But there are a lot of maybe Dr. Ramanaya can say something. He has been involved.
Ramanaya is our retired government man.
Yeah. But I think he has information on this.
Yeah, it's the NBA rules and activities are coming in a big way to transfer the technologies and the germplasm. And in the NBA, they are concerned only with value-added products other than the horticultural and crops, other crops. And there nobody understands the value of the horticultural crops or the field crops, and they don't consider them as a value-added products. And when we go there to exchange the germplasm from getting something, or even within the university, within the India university to private industry, university, they don't agree. I mean, that is a very big stumbling block, I think, as far as I can say.
Okay, I think I would sum it up that Dr. Ganeshan, I think these are areas still needing some kind of brainstorming in a smaller group to come up with suggestions. And now that we have an agriculture minister from Karnataka, I think you can try and get something energized there in Bangalore and then take it to the higher authorities. Because we cannot solve it here, we can at least bring the points to the fore. I think there are a couple of more questions, Dr. Ganeshan. Aparna, we have time?
Yes,
Aparna?
sir, we do have time.
Yeah,
You can take the questions.
So I think there are interesting questions. One is Mr. Barasahab Barpe. Can government institutions interested to work with private sector? Meaning exchange of germplasm. What would you say, Dr. Ganeshan?
Government institutions working with private sector.
Private sector.
I think this is already happening, no?
I think it is happening. It is happening actually.
AICRPs, I have seen private sector involvement. But I don't know to what extent you people think of using the germplasm, which is, see, at least I'll tell you in Australia and all, the government sector works with germplasm, the private sector works to produce varieties.
Yeah.
You can correct me if I'm wrong.
Yeah.
In India, everybody wants to work on everything. Like, private sector wants to develop varieties, private sector wants to have an R&D, without an R&D, government has to give you clearance. And in government sector, the germplasm is held by the government sector, not in terms of sharing. That proper MOU doesn't exist, and it's a long process to share the material. On the other hand, whatever material private sector brings into the government, that needs to be shared with the government, but not vice versa. These are some of the gray areas which we need to sort it out.
Another gray area that needs to have both ways. I agree. I mean, it's like
And recently they also told that material coming into the country in the form of imports, plants are being imported directly for cultivation. A set of them should be deposited to a public sector institute. This started during Dr. Krishna Kumar's
no,
period.
no, but
And
there, Ganeshan. I mean, there is a problem there. I'll tell you my personal example. Cornell University does field day every year. I have attended many of those field days. They don't send the seeds to NBPG through NBPGR.
they give direct.
We are unable to get the material because they don't. They say we are having SMTA with you as an institute, as a company, but not with NBPGR. So I think NBPGR
NBPGR. Yeah, that's true.
they need to have some way out of this. Otherwise, a lot of good material cannot come, or it will come, the in the old breeders' ways, So there are issues, Mr. Varpe. I mean, there are issues, but there are some projects already going on between private and government sector. Another Uttaranchal traders. Oh, sorry, Dr. Ramanaya. Yes, please.
Yeah, it's the foreign universities and institutions, why they don't send it through NPPR is the cause of the NPPR constitution, where NPPR constitution says whatever the material comes to them is open source to all. So the foreign institutions, they feel that they are losing IP without having any agreement with any other third party, institution, fourth institution. That NBPGR could not, it is not easy to rectify because it has to go to the parliament and get rectified. I mean, that's a big issue.
It's a very big issue because, getting inbreds that may have taken millions of dollars to develop, and then it comes to NBPGR and it becomes a national resource. It wouldn't be possible. It's like IHR sending one of their parents to, suppose, under an MTA to a company in, say, USA, under an MTA, and then US germplasm says this is a national resource. It can go to anybody. So there has to be some meeting ground on this, and I think we can't solve it here, but I think we can only flag this as an issue or future, discussion at the appropriate forum.
I see
And
Dr.
then
Karyalu,
interesting. Sorry?
I see Dr. Karyalu, former director of NBPGR.
Yes.
Maybe he may have a few words.
Dr. Karyalu, please.
Thank you very much.
Welcome.
Thank you very
welcome.
much.
Thanks for coming in.
First of all, I must congratulate Dr. Ganeshan for tackling a very, very broad topic and in such an elaborate manner. I would say the horticultural crops, by way of their life cycle and reproductive mechanisms, I say encompass a tremendously wide variety of forms, of life forms, that no crop plant other than horticultural plants, a group of crop plants can match. And in that respect, the problems with horticultural crops and that need to be solved from a scientific point of view or from a policy point of view are tremendous, are very great, and some of which we have been able to resolve, some of which we have not been able to resolve. There has been, as far as the policy issues are concerned, I am not aware of the most recent developments at NBPGR, but sincere and efforts have been made to facilitate sharing of germplasm. A committee under Dr. R.S. Paroda was constituted, and it has been liberalized to a great extent. Already there is a list under the FAO. There is a list of 60 crops that can be exchanged. That India is also exchanging. That is committed to exchange. I don't know how many horticultural crops are involved in this. For others also, efforts have been made, and I remember policy decisions have been taken that these also the exchange also needs to be pressurized. I very much appreciate the concern of suppliers of germplasm, that their germplasm should reach the intended person or the intended institute and no other institute, and which, as you say, that NBPGR says that no, it is a national asset and has to be distributed everywhere. But again, I think we need at every level we need to pursue things over and over again. There are people who, you see, these decisions have to be taken not only at the scientist level but the policy makers level also. And policy makers have so many considerations. Wherever you see, you were just talking about the political in for the import of what was that, grape germplasm. There was some political issue. So there are so many complex issues involved in that. All of us, if we make a joint effort together, I think things can look up, things can improve. I was wondering, besides from a scientific point of view, do we have, I am not aware, obvious, perhaps we may have, but I am not aware, do we have a national program on, we are talking of right now diversity, for example, do we have national programs on diversity of main indigenous horticultural crops of the country? For example, banana. Do we have a national program on diversity and genome of banana? A national genome program. A genome program would also mean the genetic diversity, molecular diversity, then other forms of morphological diversity of mango. Do we have such a program with us? Why don't we have? Who is going to have this program? And who is going to champion this program? NBPJR may do it by fits and starts, but it is the horticultural issue that has to do it. I find such programs being run by DBT, and being in those committees, I feel sorry that why doesn't any of the ICAR institutes ICAR institute become a major player in that? In some there are, but in some there are not. But as a funding agency, does ICAR or a related agricultural organization have that much commitment? I don't know because I find many of these programs are funded by DBT, not by ICAR or so. So such problems are there. Such within the system, such problems are there. I would also like to have these indigenous programs on indigenous green leafy vegetables. We talk of nutritional amelioration. Why are we stuck with only some of the, as Suren was saying, some of the exotic vegetables only? What about indigenous vegetables? Like brassicas, chinopods, amaranths, portulaca. Is there any such programs in any horticultural institute directed towards these? So quite a number of issues are there, and which we need to resolve at the institutes level, at the horticultural institutes level, at the ICAR level, as also at the policy level of the government.
Thanks for your comments.